welcome back to whatever this is. Lately I have been going through a period where everything I film I hate and I'm refilming everything 2 or 3 times, and this is one of those. I've worked quite a few scientific communication jobs, I've volunteered in museums, I've done outreach in schools, giving workshops on dinosaurs and lectures and things. I've even worked as a paleontologist in a zoo in an animatronic dinosaur exhibition, handing out knowledge and facts like candy. And there is one question that I get asked all the time. I've got a few questions that I know will come up whatever job I do, whatever age group I'm presenting to. I have a few questions that will always come up, and this is one of them. And that question is, could Jurassic Park happen? So the short answer is no, and the long answer is no. And definitely not like it happened in the book or the films. I'll go a little bit deeper with that explanation. If you've clicked on this video, I'm going to assume that you're at least somewhat familiar with the premise of the films and the book. You've probably at least seen the films, and you probably have some idea of InGen's dinosaur cloning process. But just in case you don't quite remember, let me refresh your memory. Amber-containing mosquitoes is collected. These mosquitoes fed off dinosaur blood just before death. This blood and the dino DNA contained within is extracted. The DNA is presumably amplified using some sort of PCR protocol. Any small gaps are filled in with frog DNA. This is then inserted into a viable egg in the standard cloning procedure, and blammo, a dinosaur hatches. So, first of all, let's address the short answer of no, Jurassic Park can't happen. So there's a lot of reasons for this, and I'm gonna break them down. Starting with the fact that we just cannot get dinosaur DNA. Not non-avian dinosaur. Obviously we can get dinosaur DNA because birds are dinosaurs. Not non-avian dinosaur DNA. This is the main problem with the Jurassic Park method, as DNA is such a fragile molecule. DNA in bone, it has been calculated by a study done in 2012, has a half-life of 521 years. That means by the 521 year mark, half of all that DNA that you've started with is completely unusable, or half of the bonds that hold this thing together have broken. This study was based on DNA found in the bones of moa, which is a gigantic flightless bird from New Zealand. These bones ranged from about 600 years old to 8,000 years old. And they found in this study that DNA in ideal conditions, so not just something that's randomly died over there, in ideal conditions only is readable for about 1.5 million years, and by the 6.8 million year mark it's gone. It's just completely gone. Another study in 2016 challenges this a little bit with the discovery of 1.4 million year old chloroplast DNA, which was still readable. So obviously that contradicts the other one a little bit. I don't know whether this is because it's plant DNA as opposed to vertebrate DNA, or it's because it was stored in the chloroplast as opposed to bone. There's a lot of different factors going on here that could contribute to this change, and obviously more study is needed. This study also posited the idea that after about the 100,000 year mark, which if you remember, those moa bones only went up to 80,000 years old, after the 100,000 year mark, DNA decays more slowly, and therefore you'll have a longer tailing off of readable DNA. I must admit I didn't read too much into this one because it wasn't entirely relevant to my point once it became clear that 1.4 million years is a lot more recent than 66 million years old, which is when the last dinosaurs died out. Around the time of the filming and the release of the first Jurassic Park film, there were a few studies published that claimed that they had found dinosaur DNA in Cretaceous bone, in eggshells, but these since have been very firmly proven to not be the case. Rather, it was an error in the PCR protocol, so this is a chemical reaction that allows DNA to be amplified billions of times, so you only need one single strand and then you can study it because you've amplified it so much. There were errors in this where human DNA had got into the mix and obviously replicated, so this dinosaur DNA was actually a human Y chromosome in one of these cases. Since the 2000s, no one has been able to replicate any of these results, which sort of suggests in science where you really want to replicate your results, it's probably not true. There have been a couple of exceptional cases where DNA has been collected from vertebrate samples that are very old, but in accordance with the Moa study, these only max out at about 700,000 years old. The youngest remains of non-avian dinosaurs being 66 million years old. There is a massive time gap for us to fill in with ancient DNA before we even have the slightest possibility of cloning something from that long ago. And I don't want to be a Debbie Downer, but we're never going to do it. Let's ignore all of that for a second and pretend that we did find some dino DNA locked up in maybe tooth enamel from a Cretaceous dinosaur. There are still a lot of problems with the Jurassic Park method, namely filling in the gaps in this DNA. A big one being that we would not use frogs as the gap filler. Frogs and dinosaurs haven't shared a common ancestor for a very, 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 very long time, and that makes their genomes quite different. We would instead be using birds, given that birds are dinosaurs. Even with the birds though, the last common ancestor between non-avian and avian dinosaurs was around 200 million years ago, right at the beginning of the Jurassic. So there has still been 200 million years of divergence. And that is just the ancestor with dromaeosaurs, those dinosaurs most closely related to birds. If you want to look at the divergence time between a stegosaurus and a sparrow, that would have happened at least 30 million years earlier. This is important because we want to use an animal that is as close as possible to our target animal, the one we're trying to clone and bring back to life, as possible. For the simple principle that animals more closely related share a closer genome. For example, we share around 98% of our DNA with a chimp. If you were to try and, I don't know, clone... Well, if you were to try and clone an ancient human, you would use a modern human, probably. But let's say that we couldn't do that. We'd probably use a chimp rather than, say, a banana, which we share about 40% of our genome with. Because it's no good to fill in what should be a gene for, I don't know, grow teeth with make more potassium. You're not going to end up with a viable human embryo when you do that. Another problem comes with knowing what nucleotide sequences to put in what gaps. We'd be able to sequence the genome or the remnants of whatever genome we had left quite easily. We're pretty good at doing that now. However, once we've done that, we need to figure out what everything we're looking at is and what each of these genes do and where they should go in sequence, especially if we're dealing with just little scraps. We don't know exactly if this one comes from here or if it goes over there and what goes in between. This is easier said than done when you're living in a world where making fruit flies grow legs out of their eyeballs and making glowfish is still a pretty mind-blowing thing to do. I mean, we're getting better at it, and this I imagine would be possible in the future, but not right now. Of course we have the usual problems with cloning. I mean, we're getting pretty good at cloning things that are alive or have died like in the last week. We're pretty darn good at that. There are whole businesses set up around cloning prize racehorses or beloved family pets. If you want, you can get Snoopy cloned if you have the money. But inserting the DNA of one species into the egg of another species, especially when that species is extinct already, comes up a whole bunch of new problems. As, I mean, I've discussed. There are a few projects ongoing that are looking at doing this, notably passenger pigeons, which I have my own thoughts about, but that's something for another video. Mammoths, I have thoughts again, but different video to the passengers, it's different thoughts. There are a bunch of projects that if you're interested in, all you need to do is look up de-extinction. There's a Wikipedia page that lists a lot of these projects. It's very easy to go from there and then learn more about the individual ones. The common ancestor problem is again the main problem for this, probably, because again, taking the DNA of one animal, putting it into the egg of something alive, that thing alive then also has to be the mother. So if you're dealing with a size difference or something that requires a very different in utero or in ovo environment, then there's going to be some problems there. If this is a problem between things that have incredibly recent common ancestors, like, well, I say recent, I mean relatively recent, like the 6 million years divergence time between elephants and mammoths, and that's a massive problem. There's going to be a huge problem between birds and non-avian dinosaurs. Also ethics. A lot of ethical problems in cloning. Cloned animals tend to be more susceptible to health problems. Again, I think we've mostly dealt with this with things that are cloned today, but this is going to be amplified a lot. In fact, we have managed to de-extinct an animal, and I think it lived for about 70 minutes. This was an ibex. I'll write the species name here because I have forgotten. We did manage to clone one, birth it, bring it back to life, and it immediately died. So this is another problem that needs to be overcome. And of course, looking at the ethics, is it ethical to do that to a bunch of animals so you can maybe one day have 5 that live? Okay, so Jurassic Park is off the table. No dinosaurs, never. Never happening. There could be a way to recreate dinosaurs, and that would be via genetic engineering. This brings me to the Chickenosaurus project, which is spearheaded by paleontologist and Jurassic Park consultant Jack Horner. I would take this with a grain of salt, however, since Horner has been saying, at least since 2009, that this is a project that they are going to do and they're going to get pretty quick results from. I haven't seen anything come out of it yet, besides a GoFundMe, but I reckon it will happen eventually. This project has the aim to recreate dinosaurian traits starting from the genetic base of a chicken. Chicken is used because we know a lot about it, and it would be the easiest to go into and start filling the genome. The main target areas would be turning the beak into a snout with teeth, turning the wings into more dinosaur hands, and also growing a long bony tail rather than the bird piger style and feathers. This is known as atavism activation, activating ancient traits that are probably contained within the genome. You just got to switch them on or turn off whatever's inhibiting them. If you do all of that, you get something that looks a bit like a small theropod dinosaur. There has been a group that has managed to grow these snouts with teeth on chicken embryos rather than beaks, but they never hatched them, and I think again, this is largely again to do with ethical issues. When we make these dinosaurs, we don't know what quality of life they're going to have. We don't know how best to keep them. These animals have no natural environment whatsoever. What would they eat? Would they still eat the same things as a chicken? Would they require something different? Because if we switch this gene to do this, some genes have double meanings. They might change something internally that we didn't notice when scanning the embryos. So these things could be born, be incredibly sick, and then die straight away. Which, I don't know, it's not cool. It's not cool. Another thing that is worth mentioning, especially because I've had questions on this when I've explained about the Chickenosaurus project before, this would be a completely new and artificial animal. It wouldn't have a species name per se. Maybe the species name would be Chickenosaurus, I don't know, it'd probably be more like Galosaurus, something like that, using the Latin, but for now it's Chickenosaurus. This would not be something like Velociraptor or Deinonychus. It wouldn't be an already existing animal because those are dead and gone. They are not coming back. This would be new. There are reasons to undertake this project besides dinosaurs are cool, I want one. Probably the main one of these is to do with the implications for human spinal research that doing the research into growing the long bony tail could have. We could find something that could be very useful for medical research whilst doing that in the genome. I won't go into all the reasons, instead I will direct you to Horner's book, How to Build a Dinosaur. This is a pretty good read. It outlines everything in the project, everything it could be useful for, a little bit of background as well, some stories from Horner and his lab, like Mary Schweitzer, the work they've been doing. It's all very interesting, it's a good read, even if it does read a little bit like Horner's personal grant proposal and almost reasoning to himself to why he should be allowed to do it, which it is. But apart from that, it is a good read anyway, and I would recommend it. I will also link down below to a TED talk that he gave on the subject, which isn't in nearly as much detail, but it does mention the project, and it's probably worth a watch. Obviously, being a TED talk, it's less than 18 minutes long, so it's not going to take up a long period of your time. Well, there you go. The answer to could Jurassic Park happen is no. Asterisk. If you have any other burning paleontological questions, please feel free to ask them in the comments below, and I might make a video on them. I might ignore them, but I might make a video on them. Like Jack Corner, I'm going to do whatever I want. Thank you for watching, I hope you enjoyed the video, and I hope you learnt a little something. And I will see you in the next one. There's nothing in this mug. I just wanted to show off my birthday present mug. Got dinosaurs on it.